Good afternoon, class. I am Miss Lamar, and today we're going to be talking about cellular respiration, and we're going to start the class a little differently. So if everyone will stand up, you are going to show me your best dance moves and get as most energy out as you can for about 30 seconds, maybe a minute. The music's in there, you know. Keep yourself up.
We're not going to do a video for us?
Alright. So, saw some good dance moves from Lindy. That was really good. Yeah.
She's got some groove, doesn't she?
Does dancing or moving a lot, does it make you out of breath?
Mm-hmm.
Or like when you're running? Does it make you out of breath and wondering? So like, why do you get out of breath when you're really active and using energy? Have you ever thought about it? Why do you always have to breathe heavy after you've just done a workout, or maybe you play sports and you have to go run track or something? You ever thought about that?
Because you're using your muscles so much and you're exercising so hard.
Yeah. So you're breathing in a lot because you're trying to get oxygen, because our cells use oxygen as fuel. And so we receive oxygen through a process as breathing, which is also known as respiration. So knowing that respiration is you breathing, what do you think cellular respiration might be?
Cells breathing.
Cells breathing. Okay, any other guesses?
Cells taking in oxygen.
Okay, yeah, that's good. Okay, so we're going to learn about that today. So this is a chemical formula for cellular respiration. You might notice it looks very similar to photosynthesis. It is actually the opposite as photosynthesis. So if you remember in photosynthesis, the autotrophs or plants, for example, they can use the carbon dioxide in the air and then sunlight, and they can produce their own glucose and oxygen. And they release the oxygen that we breathe. And so we are heterotrophs, meaning that we have to take in that glucose and that oxygen from another source. We can't create it ourselves like plants can. And so we need oxygen and glucose to get energy. And so we get this energy through cellular respiration. And so cellular respiration comes in 3 stages. So here's just a quick overview of what that is. The first stage is glycolysis, which takes place in the cytoplasm. And then the second step is the Krebs cycle. And then it goes into the mitochondria. And then the final step is the electron transport chain, which is also in the mitochondria. And so this cellular respiration is the process of making energy in the form of ATP. And so ATP stands for adenosine triphosphate, and it's commonly referred to as the currency of the cell, or the money of the cell. So like, say you need to go, you're going to go to Walmart and buy yourself a new top or something. You have to pay for that, right? To get something you have to pay for it, but you can't just pay with anything. You can't pay with monopoly money. You can't even pay with money from England, like a yen for China. You know, you have to use the right currency of money. And so the currency of the cell is ATP. It's adenosine triphosphate. And so that is made up of an adenine group, a ribose, and 3 phosphates. And so that's where the hint triphosphate comes from, is that you have 3 phosphates. And so when one of these bonds, when you lose one of the phosphates, it releases energy, which is what the cell needs. And you need ATP for muscle contractions. So like when we were dancing and you were moving around, your muscles needed ATP to be able to contract. Even when you're sitting down now, just listening, you need ATP to be able to breathe and to exhale, to move, to write down notes. So all of that requires ATP, which is why cellular respiration is so vital. So the first step is glycolysis, and as I mentioned earlier, it does take place in the cytoplasm. The main steps of glycolysis is you have glucose from the food you eat, and you're trying to get to pyruvate. And it looks really complicated. There's a total of 10 steps in glycolysis. We're not going to go into what those steps are today, but you just need to know that it is the conversion of glucose to pyruvate. And it's also really important to note that it requires ATP to start glycolysis. So in this chart up here, you can see you have a molecule of glucose and you're trying to get to pyruvate. However, you have to use 2 ATPs to break down glucose. And so in order to break down glucose through a series of steps, you are required to give up 2 ATPs. So it's kind of requiring energy to make more energy. And so once glucose is broken down through a series of reactions, which enzymes play a very key role in, you get to a step where energy is now being made. And so you get an NADH, which carries electrons. It's kind of like a car carrying you to where you want to go. Electrons need to go to the electron transport chain, and NADH is like the vehicle that can carry those electrons. And you also get 2 molecules of ATP. And because glucose is broken down into two pieces, this process happens twice. So you have 4 ATP made. And so you have a total of 4 ATP being produced for 1 molecule of glucose. However, you use 2 in the first step. So it's 2 minus 4, which equals
2.
2. So you have a net, it's called a net charge, of 2 ATP produced. So even though you gave up energy in the first step, you got it back and you doubled it. So the next step and then glycolysis has recently been helped in diagnosing cancer, because in cancer cells, they like to divide and they're growing out of control, and that requires a lot of energy. And so glycolysis can be found as if you have a lot of cells needing glycolysis or glucose, which is the first product of glycolysis, it's normally a sign of these cells are really wanting to create energy because they're growing and dividing. And so PET scans can be used to see if cells are using more glucose than normal, such as this picture here. This is a picture of a PET scan, and it's showing really high levels of glucose. It's actually it looks like the liver, but you can't really see in the picture, in the liver. And so this patient has liver cancer, and it was detected through a PET scan, indicating high levels of glucose.
What does PET mean?
It is, I forgot the name.
It's not like PET.
Oh, no, it's not like a PET. It's a medical device used to detect diseases such as cancer. I could look up the real name.
It's okay.
It's an imaging equipment used by the medical professions. So the second step of cellular respiration is the Krebs cycle. It's also known as the citric acid cycle. And this takes place in the mitochondrial matrix. So the pyruvate that was formed in glycolysis is now used to start the citric acid cycle with acetyl-CoA. And again, it's a long, complicated process. You just need to know that in the citric acid cycle, you also are creating more NADHs, which can carry the electrons, and FADH2, which are also used similar process as NADH. And so this whole process of glycolysis and now the Krebs is to make ATP and make energy for your cell, because all of your cells require it for everything. So, and the final step, the electron transport chain, it takes place in the inner membrane of the mitochondria. And it is composed of 4 protein complexes, and the final one is kind of like the main one, and it's called the APT synthase. And in the past few cycles, we've made a couple of ATP, 2 from glycolysis. We have the factors for ATP in the Krebs cycle, but that's not, we haven't really made a lot of ATP at this point. We've made some, but not a whole lot. And that's where this complex comes in. And it can make up to 38 APT per 1 molecule of glucose. And so that's a lot of ATP for 1 molecule of glucose. And when you eat food, you get a lot more than 1 molecule. So just think of like, this is really producing a lot of energy for your cells, a lot of energy for you to use. And so this can kind of be a hard concept to grasp. So we're going to watch a quick little video just demonstrating it, so that way you have a visualization of what goes on in the cell during the electron transport chain.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron 1 at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron
It's just
donation.
a good visual to see what's actually happening with the electrons, because it can be difficult to visualize. In Genesis 2:7, it states that, and the Lord God formed man of the dust of the ground and breathed into his nostrils the breath of life, and that man became a living soul. And so when God breathed life into Adam, he became living. It's that breath of life became living. And up until this point, it's talking about the creation at this point. Everything that was needed for us to live and to survive and to function was created right before everything. So like I said, like with the glucose, we need that. We need to take that in. We're not like plants who can make it ourselves. So that was created, and then all the animals were created, and then we were created then. And so through that breath of life and through the oxygen, we became living like Adam. And so do have an activity just to see this test. It is worth points. It's worth 15 points and it will be part of your study guide. It's the main key terms of this lesson. And so we'll continue on with cellular respiration the next couple of days just to go over what the actual steps involve, what's all entailed. And so there's questions.